Hi everybody, and welcome to our module on nephron physiology. Shown on the screen is a drawing of the nephron. You can find millions of these in the human kidneys, and the job of the nephron is to filter the blood and make urine that will be excreted from the body, basically to take some elements out of the blood and excrete them in the form of urine and leave others behind. And the first basic principle of how the nephron works that you need to understand is that the nephron filters a lot more stuff than it wants to excrete in the urine. So blood comes into the nephron here at the glomerulus, and lots of things, water and other solutes, are filtered that the body actually wants to hold on to. And what's then going to happen is, as those substances move through the nephron, they are going to be selectively reabsorbed. So for example, things that the body wants to hang on to, like water and sodium chloride and potassium and all these things I've shown here, will be reabsorbed. And you can imagine how this evolved. Millions of years ago, our ancestors just had a filter, and lots of things that were useful to the body went out in the urine. And then over time, the nephron evolved to selectively reabsorb the useful substances, and now we're able to pick those back up and hang on to them. The way that the nephron selectively reabsorbs solutes and other substances that are useful to the body is by transporting them from the urine side of the nephron into the interstitium or blood. There are cells that line the tubules of the nephron, and they perform this job of transporting solutes out of the urine and into the interstitium or blood. First thing you need to understand about how this works is that these cells that line the nephron are polarized. They have one side called the apical membrane. That's the side that faces the urine or the lumen of the nephron. And they have another side called the basolateral membrane. That is the side of the cell that faces the interstitium or blood. So first let's start by talking about how a substance like sodium might be reabsorbed. And let's imagine that we have a very high concentration of sodium in the urine and a relatively low concentration of sodium inside a cell lining the tubule. If this is the case, sometimes the nephron will reabsorb a solute like this by having a protein that assists and simply grabs the sodium from the high concentration side and moves it to the low concentration side. This does not require energy because we are going from a high concentration to a low concentration. On the other hand, if we have a situation where there's a relatively lower concentration of sodium in the lumen and a relatively higher concentration inside the cell, then we can also have a transport protein that assists in this process, but that will require ATP. It will require energy because we are going against the concentration gradient. Sometimes the tubule cells in the nephron won't use any proteins at all. They'll just use simple channels. So if we have a high concentration of sodium in the urine and we have a low concentration in the interstitium of blood, then sometimes cells will just have channels or openings where sodium can diffuse in and diffuse out. And this is another way that reabsorption of solutes can occur. And then finally, in some cases, the cell won't be involved at all, but sodium and other solutes will simply be absorbed by what's known as the paracellular route. This can happen if we've got a low concentration over here and a high concentration over here. Then solutes can just pass through the spaces between cells and be reabsorbed by the kidneys. Osmotic diffusion is another method of transport used in the nephron that is unique to water. So for example, if we have solutes dissolved in the urine side of the nephron such that we create a low osmolarity of 50 milliosms, and if we have a high osmolarity of say 1200 milliosms in the interstitium or blood side, then water can be reabsorbed by osmosis. It can simply diffuse into the cell and out of the cell, or it can be reabsorbed by the paracellular route. The idea here is that osmosis is a unique process to water, and anytime we create a difference in osmolarity between the lumen and the interstitium or blood, we can then reabsorb water by osmosis. Now let's talk about the segments of the nephron. So blood enters here in this portion of my drawing called the glomerulus. And remember what I told you before, lots of things are filtered at the glomerulus that the body does not want to lose in the urine, and these things will be reabsorbed as fluid moves along the tubule of the nephron. In addition, some segments of the nephron can secrete substances into the urine. You can basically think of the nephron as 5 different segments, each doing a different job in terms of reabsorption or secretion. The first segment is the proximal tubule, then we have the descending limb of Henle, then we have the ascending limb, then we have the distal tubule, and finally the collecting duct. And in this video, we're going to go through these segments one at a time and talk about what they do. We'll start by talking about the proximal tubule. I like to think of this section as the workhorse of the nephron. It reabsorbs more solutes than any other portion of the nephron, and we'll talk about this as we go along. First of all, this section of the nephron reabsorbs 100% of glucose and amino acids under normal circumstances. These are very useful solutes to the body. Glucose obviously is an energy source, and amino acids are important for protein synthesis. The body does not want to lose these in the urine, so any that get into the nephron through the glomerulus are going to be completely reabsorbed under the proximal tubule under normal circumstances. In addition, other solutes that are helpful to the body, things like bicarb, sodium chloride, potassium phosphate, and also water, are reabsorbed in very large amounts at the proximal tubule. 2/3 of these substances are reabsorbed. So basically, right after the filter, the glomerulus, when all these things enter into the nephron fluid, immediately 2/3 of them are reabsorbed by the proximal tubule. The engine that drives the proximal tubule is a sodium-potassium ATPase pump found in the basolateral side of cells of the proximal tubule. It's very important that you understand this. This pump makes the proximal tubule work. It is always going, consuming ATP. The proximal tubule uses a lot of ATP, and this creates a low concentration of sodium inside cells in the proximal tubule. And what the cells are going to then do is use this low concentration of sodium as a driving force to reabsorb lots of other solutes and also water. As I told you before, proximal tubule cells reabsorb all of the filtered glucose under normal circumstances. Once there's a low concentration of sodium inside the cell, this is very easy to do. This creates a situation where we have a relatively high concentration of sodium in the lumen or the urine, and therefore sodium can diffuse down its concentration gradient linked to a pump which also reabsorbs glucose. And then there is another pump on the other side of the cells, and in this manner glucose is 100% reabsorbed from the urine to the interstitium or blood. All that potassium that is put inside the cells, creating a high concentration of potassium, can then leave the cells linked to chloride, and this pulls chloride out from inside the cell, and then chloride reabsorption can be linked to the excretion of anions. Anions are negatively charged substances. They have a tough time getting across the glomerular capillary membranes, and they often don't get filtered very well. So the proximal tubule cells need to secrete these substances to get them into the urine. So things like hydroxide, formate, oxalate, sulfate, these are all negatively charged molecules, and they are secreted by the proximal tubule cells linked to chloride, and the reason chloride can be reabsorbed is once again because that ATP pump is always working. And then once we get a high concentration of sodium over here from reabsorption, then we can use osmosis to reabsorb water via the paracellular route, and some of that water takes sodium chloride with it. So in this manner, we grab more water and sodium chloride from the lumen or the urine and bring it back into the interstitium or blood. Let me pause and make some important points about glucose. It's very high yield for you to understand this. As I told you before, this is completely reabsorbed by the proximal tubule under normal circumstances, and it's reabsorbed together with sodium by a co-transporter. If a patient develops a high glucose concentration in the serum, as occurs in diabetes, if the glucose level gets to about 160, then glucose will start to appear in the urine. At 350 milligrams per deciliter in the serum, all transporters are saturated, and at that point, any further increase in serum glucose leads to more glucose in the urine. If we were to plot the serum concentration of glucose on the x-axis, and if we were to plot the urinary excretion of glucose on the y-axis, what we would see is initially this would just be a flat line at 0. The reason it would be a flat line at 0 is because there's no urinary excretion of glucose until the serum glucose concentration rises to about 160. At that point, we would start to see a curve upwards as some of the proximal tubule cells become saturated and are unable to reabsorb any further glucose. Once we hit about 300 milligrams per deciliter, we would then see that this would become a straight line at a 45-degree angle. And the reason for this is because at this point, every reabsorbing transporter in the nephrons is saturated, and anytime the serum concentration goes up any further, it just increases the amount that's being excreted in the urine. And you may know that diabetes mellitus gets its name because the urine in this condition is sweet. Hundreds of years ago, people tasted it, and the reason it's sweet is because of the glucose. It is very, very high yield to know that in pregnancy, some glucose in the urine or glycosuria is normal. The reason for this is because the GFR goes up in pregnancy as the plasma volume goes up. In addition, there is evidence that there's some decrease in glucose reabsorption. And for this reason, if you want to screen mothers for diabetes occurring in pregnancy, you need to use serum glucose testing, because finding some glucose in the urine doesn't necessarily mean the pregnancy is abnormal. Let me also make a special note about amino acid reabsorption in the proximal tubule. Just like with glucose, all amino acids are reabsorbed in the proximal tubule under normal conditions. Just like with glucose, they are reabsorbed together with sodium via a cotransporter, as I've shown at the bottom of this slide. There's a clinical tie-in here with a rare disorder called Hartnup disease. Children born with this rare disorder have no tryptophan transporter in the proximal tubule. Tryptophan is an amino acid, so as a result, they lose tryptophan in the urine and they develop a tryptophan deficiency. Tryptophan is an amino acid used to synthesize the vitamin niacin, so children deficient in tryptophan from Hartnup disease will develop niacin deficiency, and niacin deficiency has a skin rash associated with it called pellagra. Pellagra consists of plaques on the skin and desquamation. So the way this will present in a board question is a baby who develops a skin rash in sun-exposed areas and then has amino acids in the urine. Remember, you should never see amino acids in the urine under normal circumstances. If you see this together with a skin rash in a baby, you should think of Hartnup disease. The problem is amino acid reabsorption in the proximal tubule of the kidneys. Now let's talk about the reabsorption of bicarbonate in the proximal tubule. There are some metabolic processes in the body that produce H+ or acid. There are other metabolic processes in the body that produce bicarbonate or HCO3 negative. However, on balance, normal metabolism produces acid every day, and we need bicarbonate around to buffer this. And as I talk about in some of the acid-base videos, our kidneys can actually synthesize bicarbonate, but before they go to all the trouble of making new bicarbonate, they want to make sure they reabsorb all the bicarbonate they can, and therefore reabsorption of bicarbonate in the proximal tubule is very important. The way this is done in the proximal tubule is through an antiporter of sodium and hydrogen ions. These hydrogen ions go out into the lumen or the urine where they combine with bicarbonate to form H2CO3. There is then an enzyme that is present in the kidneys called carbonic anhydrase, a very important enzyme, which can convert H2CO3 into carbon dioxide and water, which can then enter cells in the proximal tubule. The same enzyme, carbonic anhydrase, inside of cells can then reform H2CO3, which can then break apart into another H+ and an HCO3 negative atom. The HCO3 can then be carried out into the interstitium and blood together with sodium, and the hydrogen ion is recycled to keep this whole process going in a loop. And the net result of this whole process is that HCO3 is reabsorbed into the interstitium of blood, and it takes with it sodium, so that sodium reabsorption is linked to bicarbonate reabsorption. In addition to bicarbonate being important for acid-base balance, there are a number of other clinical correlations of bicarbonate reabsorption that I talk about in other videos. In the diuretic video, I talk about carbonic anhydrase inhibitors. These are weak diuretics because they block sodium reabsorption. They also lead to bicarbonate loss in the urine. And in the renal tubular acidosis video, I talk about a type 2 RTA, which is an ion defect where patients have the inability to absorb bicarb in the proximal tubule, and this leads to a metabolic acidosis. Fanconi syndrome is relatively rare, but it is very high yield for you to understand this syndrome for your boards because the cause of Fanconi syndrome is a loss of all of the proximal tubule functions. Basically, in this syndrome, the proximal tubule doesn't work. As a result, you see impaired resorption of solutes. Remember, as we have discussed, the proximal tubule is critical to reabsorbing many solutes that the body wants to reclaim. This includes bicarbonate, glucose, amino acids, phosphates, even some small low molecular weight proteins which can be filtered at the glomerulus. They are reabsorbed by the proximal tubule, and all of these things are going to go out in the urine in Fanconi syndrome, and they like to test you on this to see if you understand what the proximal tubule is supposed to do. The presentation of Fanconi syndrome is very similar to diabetes. That's because there is glucose in the urine, which leads to diuresis. This makes the patient urinate a lot, and then the patient becomes thirsty, so there will be polyuria and polydipsia. And in a board question, you will see a description of polyuria and polydipsia along with glucose in the urine, and you will say, aha, this is diabetes, but it's not. And the reason is because in Fanconi syndrome, there is a normal serum glucose. There is no hyperglycemia because insulin and glucagon and all the body systems to control glucose levels are working. You can also see a non-anion gap acidosis. This is because you're losing bicarb in the urine. There can be hypokalemia because anytime there is increased nephron flow, this leads to a loss of potassium, and we'll talk more about this later in this video. There can also be hypophosphatemia from loss of phosphate, and there can also be amino acids in the urine. And remember what I told you before, in Hartnup disease, amino acids should never be in the urine, and if you see a description of this, you should automatically think something is wrong with the proximal tubule. Fanconi syndrome can be inherited or acquired, and as I told you before, in general, it's rare. The inherited form, which presents in infancy, is associated with cystinosis. This is a very rare lysosomal storage disease where babies cannot break down cysteine and therefore it accumulates in their cells. If you look at the bottom of the screen, this is the amino acid cysteine. If you link two of these together, you get cysteine. And cysteine often links together to form this structure of cysteine, and you need to be able to break this down in your lysosomes. If you're missing an enzyme,